Good afternoon, children.
Good
We were
afternoon,
learning
ma'am.
about regulation of gene expression in the previous period, and we started with lac operon. Now we are going to learn in detail about lac operon. And operon, as I told you, it is an arrangement where a polycystronic gene. We have learned about monocystronic gene and polycystronic gene. Polycystronic gene codes for more than one polypeptide. So operon is an arrangement where a polycystronic gene is regulated by a common promoter and regulatory genes. Or else you can say it is a functional unit of DNA containing a cluster of genes all under the control of a single promoter. Now, there are different types of operon, and each operon is having its own specific repressor protein. About repressor protein, I told you repressor protein will bind with operator. Repressor protein, they will bind with operator. What are operators? Definite sequences of DNA lying close to the promoter. And once repressor protein binds with the operator, our polymerase enzyme will not have access to promoter. And then the process cannot take place. So we are going to learn in detail about lac operon. It's a very important topic. All these we had seen. The elucidation of lac operon as a transcriptionally regulated system was first done by geneticist Francois Jacob and biochemist Jacques Monod. So, lac operon was explained as a transcriptionally regulated system by two scientists, Francois Jacob and Jacques Monod. And let's see what are the different genes involved here. All these genes are important part of lac operon. So as such, operon means it's a definite sequence of DNA where several genes are under the control of a common promoter. So these are the genes involved here. I gene, Z gene, Y gene, and also A gene. A gene, Y gene, Z gene, and I gene. I gene acts as the inhibitor. I gene is the inhibitor because we call it as an inhibitor because this particular gene is the one which codes for repressor of lac operon. That is coding for, you know that each gene, the proteins are formed as a result of the expression of gene. So I gene, Z gene, Y gene, A gene. I gene, we can even call it as inhibitor gene. Remaining all of them are structural genes. You call it as inhibitor gene because this codes for repressor protein. You know, repressor protein will inhibit the transcription process. Now, Z gene, it codes for beta galactosidase. So we are discussing about lac operon. Lac stands for lactose. So the Z gene codes for the enzyme beta galactosidase. And what is the role of this enzyme? It will convert lactose into galactose and glucose. Both of them are energy sources for E. coli bacteria. Then Y gene, that is also structural gene, and it codes for the enzyme permease. All are proteins, repressor protein, beta galactosidase, that enzyme is protein, permease and transacetylase. Both of them are proteins. And permease, its function is to increase the permeability of the cell to lactose. If lactose should enter the cell, then this enzyme is needed. Only in the presence of this particular enzyme, lactose can enter the cell, or the cell becomes permeable to lactose. So only if lactose has entered, there will be the need for beta galactosidase enzyme, and once it is there, it will be converted to galactose and glucose and provides energy to the bacteria. And A gene codes for the enzyme transacetylase. Its function is not mentioned in your textbook. Its function is to protect the cell from the building up of toxic proteins. So transacetylase enzyme will protect the cell from the building up of toxic proteins. So these are the different genes that constitute lactoferrin. All the genes are equally important. I'll repeat once again: I gene, Z gene, Y gene, A gene. IZYA. I gene is inhibitor because it codes for the repressor protein. Repressor protein can bind with operator and prevent the transcription process. So we call it as this gene which is coding for repressor protein as inhibitor. Then we have Z gene. Z gene codes for the enzyme beta galactosidase, which will hydrolyze or which will convert lactose into galactose and glucose. Then Y gene. Y gene codes for the enzyme permease. Why permease is essential? This enzyme will promote the lactose, promote the cell to absorb or take in lactose. Or if this enzyme is not there, cells will be impermeable to lactose. Then A gene codes for transacetylase. It is necessary to prevent the building up of toxic proteins within the cell. Have you got the different names of different genes and their function?
Yes.
Okay, I'll change. I think you have finished it. It's very important, children. Role of each genes. All genes, all the genes involved in lactoferrin are required for the metabolism of lactose. So all the genes are equally important. So lactoferrin is concerned with the metabolism of lactose. So all the genes which I mentioned, they are equally important for carrying out the lactose metabolism. And lactose, we call it as inducer. Lactose molecule is known as inducer. Inhibitor is the I gene because it codes for the repressor protein. Whereas lactose you call as inducer because lactose is a substrate for beta galactosidase. And the presence of lactose regulates switching on and off of the lactoferrin, hence it is called inducer. So last period I had told that if the substrate is not there, there is no need for the production of the enzyme. So if lactose is not there, there is no need for the production of beta galactosidase enzyme. So the gene responsible for beta galactosidase need not get expressed. But if lactose is there, beta galactosidase is needed. If lactose is absent, the enzyme is not needed. So since the presence of lactose is inducing the formation of beta galactosidase, you can call it as inducer because only in the presence. See, this lactoferrin is concerned with the metabolism of lactose. And when lactose is present, the process will take place. So lactose molecule can be considered as the inducer because lactose is a substrate upon which beta galactosidase acts. So the presence of lactose can result in the switching on and off of lactoferrin. When lactose is not there, this can get turned off temporarily, and when it is present, it can get turned on. So you call it as inducer molecule. If lactose is provided to the growth medium of bacteria in the absence of any other carbon source like glucose, then it is transported inside the cells by permease. Already I told you the role of permease. So when the cells are provided or these bacterial cells are grown in a medium containing lactose and no other carbon source, like no other molecules with carbon like glucose are present, then at the same time if lactose is present, through this cell with the help of the enzyme permease, lactose will be taken up, resulting in the turning on of lactoferrin. So lactose is a source of energy. Lactose, once it is broken down to glucose and galactose, that will be the source of energy for the bacteria. And when there are no other carbon source like glucose, and if lactose is present in the absence of other carbon sources, it will be taken up by the cells in presence of the enzyme permease. Okay, we'll come back to all these points later. For permease to be present, And for the lactose to enter, a low level of expression of lac-operon must be present all the time. A very low level of lac-operon. It should not get completely shut off. A low level of expression of lac-operon should be maintained throughout, because only then the permease can be present, and with the help of this permease, lactose can enter. So a low level of expression is very important. Otherwise, lactose can no longer enter and it will remain shut off, because only when lactose is there, the system will become active. Only when lactose is present, beta galactosidase will be produced and it can act upon this. So always a low level of expression of lac-operon is needed so that lactose can be absorbed in presence of permease. Now let's see how the regulation is taking place. If you haven't understood the points that we mentioned, not to worry, we'll go back to all the slides under lac-operon once I finish it. Regulation in the absence of inducer. Just imagine that we don't have inducer. Lactose is not there. Okay, then what will happen? How the gene expression is regulated. So here you can see the different genes: I gene, Z gene, Y gene, A gene. All the genes are, see, I, Z, Y, A. Here you can see the repressor protein. How is repressor protein formed? Is the I gene. First, it will be transcribed. The gene will get transcribed into messenger RNA. Then they will get translated into proteins. That is how proteins are formed. Whether it is repressor or whichever protein it is, they are formed in this way. Once again, I will repeat. Here, no inducer. So, we will be seeing how the gene expression is regulated in presence and in absence of, in presence and also in the absence of inducer. So first we are going to see how the gene expression is regulated in the absence of an inducer, in the absence of lactose. And I gene, this is I gene. I gene, first it will get transcribed to form repressor messenger RNA, then it will get translated. This is transcription. This is translation to form, translated to form repressor protein. Okay. Then this repressor protein will go and bind with the operator. So it will go and get attached with the operator. See here you can see it will bind with the operator. Operator, I told, lying close to the promoter. It's a sequence of DNA. So once the repressor protein is formed, they'll go and bind with the operator. Then what will happen? Once this has happened, now this will prevent RNA polymerase from recognizing or from binding with the promoter. And if that cannot happen, if RNA polymerase, for the process to begin, for the initiation to happen, RNA polymerase, DNA dependent, in transcription you had learned, DNA dependent RNA polymerase in presence of the initiation factor should bind with the promoter for the process to start. But here repressor protein has gone and attached with the operator. So in this situation, the RNA polymerase will not have access to the promoter. So as a result, what will happen? Z gene, neither the Z gene, Y gene, A gene can be transcribed. Actually, Z gene will form they all will form their respective RNAs, and then they will get transcribed into, they'll get translated into respective proteins, beta galactosidase, permease, transacetylase. All that will not happen because here there is no inducer. Okay, I told you there is no need for the formation of these enzymes if lactose is not there. That is, this is how the gene expression is regulated. Now, lactose is not there, so it is not needed. The enzymes beta galactosidase, permease, and transacetylase are not needed. So this is how it gets regulated. Regulation of lac operon by repressor is known as negative regulation. Therefore, in the absence of the products of these genes means, what are the products of these genes? Z gene. Z gene will produce beta galactosidase. Y gene permease. A gene transacetylase. So when the inducer is not there, the products of these genes, that means the different enzymes, cannot be produced, and thereby metabolism of lactose cannot take place. Because if lactose is not there, actually these enzymes are not needed. Only when lactose is there, we need the enzymes to break it down. So this is how the regulation is taking place in the absence of inducer. I'll repeat once again. Regulation of lac operon in the absence of inducer. Inducer molecule is lactose, because lactose will only in the presence of lactose beta galactosidase is needed, and the lac operon will turn off and on depending upon the presence of lactose. So when lactose is not there, I gene will get transcribed and form repressor messenger RNA, then it will form repressor protein. Lactose is not there, and this repressor will go and bind with the operator. Once this has bound with the operator sequence, the RNA polymerase cannot, or they will not get access to the promoter. Only if the RNA polymerase has got attached here, the further steps can take place. So since RNA polymerase cannot initiate the process, the transcription of Z, Y, and A genes will not take place, and the proteins, the enzymes will not be formed. Regulation. So this method or this way of regulating lac operon by repressor is negative regulation. And when these enzymes are not produced, the metabolism of lactose can no longer take place. Now let's see what happens if lactose is present. When lactose is present, we need the enzymes to break it down. Like beta galactosidase is needed, permease is needed, transacetylase is needed. So all these enzymes are needed when lactose is present. Now, how does that happen in the presence of lactose? So regulation in the presence of inducer. Inducer is lactose. So when lactose is present, here also I gene will get transcribed. I gene will be transcribed to form repressor messenger RNA, and they will get translated to form repressor protein. But when lactose is there, what it does? This lactose will combine with the repressor protein and makes it inactive. Repressor is made inactive when lactose is present. If lactose is not there, once the repressor protein is formed, they'll go bind with the operator. But when the repressor protein or when the repressor protein is produced in presence of lactose, this lactose will go and bind with the repressor protein and makes it inactive. Once it has become inactive, they cannot mask this site or they cannot bind with the operator. So the promoter and operator sequences are free. Now RNA polymerase can bind with the promoter and the process can be initiated, and all these will get translated into their respective first messenger RNA will be formed, and then they get translated into the respective enzymes. Beta galactosidase, Y gene permease, A gene transacetylase, and the process will go on smoothly. Lactose metabolism will take place smoothly. This inactivated repressor will be unable to inactivate RNA polymerase. And as a result, the enzyme access, that is enzyme means RNA polymerase, they can access the promoter region and transcription will proceed. Z, Y, and A genes will synthesize their respective messenger RNA, which will get translated into the respective proteins, beta galactosidase, permease, and transacetylase. In presence of all these enzymes, the metabolism of lactose will proceed in a normal manner. Okay, I will repeat from the beginning of the lac operon quickly. From lac operon, I will start. Operon is an arrangement when a polycistronic gene is regulated by a common promoter and regulatory genes, or else you can say it's a functional unit of DNA with a cluster of genes under the control of a single promoter. There are different operons depending upon it is concerned with the metabolism of which substance or which molecule. Lacoperon, tripoperon, histoperon, tryptophan, histamine, valine. Okay, and here we are going to learn. You need to learn only about lac operon in detail, and this particular operon was explained as a transcriptionally regulated system by two scientists, Francois Jacob and Jacques Monod. You should know the names. Just the names of the scientists can be asked, and you will be asked to list their contribution. So the different enzymes involved are I, Z, Y, and A gene. I gene can also be called as inhibitor because this one codes for repressor protein. Each gene will code for a particular protein. So it is coding for repressor protein. Z gene for beta galactosidase, which will help in breaking down lactose into galactose and glucose. Y gene codes for permease, which will help in increasing the permeability of the cell to lactose. So if lactose of this sugar should enter the cell should be permeable to that. And for the cell to become permeable for that, the enzyme permease is needed. And finally, A gene, it codes for transacetylase. It will prevent the building up of toxic proteins within the cell. And all genes, we learned about 4 genes, all of them are important to carry out the metabolism of lactose. And out of the different substances that we discussed, lactose acts as the inducer molecule because that decides whether the lac operon should take place or whether the metabolism should take place or not. Because only when lactose is there, beta galactosidase should be produced to break it down into glucose and galactose. So it is a substrate for beta galactosidase, and thereby it regulates the switching on and off of lac operon. I told you when there is no lactose, when there is no substrate, we don't need any enzyme. Beta galactosidase should be produced when there is lactose. If lactose is not there to break down, there is no need of beta galactosidase. So thereby lactose acts as the inducer. And if lactose is given to is provided in the growth medium in the absence of other sugars or in the absence of other carbon source, it will be taken up by the cells with the help of the enzyme permease. Always there should be a low level of lac operon so that the lactose can be absorbed and the permease can be present to facilitate the same. And then we learn how the lac operon works in the presence and absence of inducer. To begin with, I gene will transcribe to form the repressor messenger RNA. It will get translated to form repressor protein. This repressor protein will bind with operator. Hope you remember what I told about operator. Operator is a sequence of DNA which is lying close to promoter. And once the repressor protein binds with operator, it will prevent the transcription process. And why it is preventing the transcription? Once the repressor protein has attached to the operator, the enzyme to start the process, initiation can take place only when RNA polymerase binds with the promoter region in presence of initiation factor. But when the operator sequence or when the operator region is having this repressor protein bound to it, the RNA polymerase cannot get access to RNA polymerase, and thereby the transcription of Z, Y, and A gene cannot take place. So no lactose, no enzymes are produced. The repressor binds with the operator of operon and prevents RNA polymerase from transcribing the genes. And regulation of lac operon by repressor is negative regulation. So in the absence of these enzymes, products of genes means all these genes are getting transcribed to form enzymes. So in the absence of these enzymes, which are the products of genes, each gene should get expressed to form the respective protein. So when the products are not formed, metabolism of lactose cannot take place. But if lactose is present, in presence of inducer, what will happen? The same process, repressor protein will be formed by the I gene. I gene will get expressed and they will form repressor protein. But this repressor protein will be inactivated. In the second case, in the previous one, there was no inducer or lactose. But here we have lactose. This lactose will bind with the repressor protein once it has formed and it will inactivate it. So once inactivated, repressor protein cannot bind to the operator, and the promoter region lying next to the operator is free for binding with RNA polymerase. So RNA polymerase will bind with that and it will initiate the process, and all the respective messenger RNA and the proteins will be formed and the process will go on smoothly. Those who attended the last class, have you followed? Punya, not very sure. Understood?
Yes,
Understood. What about others who did not understand? You can raise your hands. I will repeat. Have you understood? Nandana? No. Okay, who else? No problem. Don't hesitate to say no. Who did not understand? Nandana, where did you not understand? Tell me. Which part? Will you message me? Because I'm not able to hear your voice. Just message me. Absence of inducer. Okay. That is easy to understand. Wait. Okay. Absence of inducer. What is inducer, Nandana? Which is the inducer molecule? Nandana, which is the inducer molecule? What acts as the inducer?
lactose.
You don't have to unmute because it doesn't make any change. You can just say lactose. Lactose is the inducer. Okay. And inducer is absent. There is no inducer or there are no lactose molecules. So here I gene's function, it will perform. I gene will get transcribed and form its messenger RNA, and then translation takes place. Repressor protein will be formed. And once the repressor protein is formed, it will bind with the operator. Once it is attached with the operator, RNA polymerase, which has to initiate the process, they will not have access to this promoter. It is prevented from accessing the promoter region because the repressor protein has already occupied the region next to the promoter. But when lactose is there, what the lactose will do, that lactose molecule, that is in presence of inducer, the lactose molecule will bind with the repressor and inactivate it. Once inactivated, it cannot bind with the operator. So it is free. So the operator region and the promoter region, they are free. There are no repressor proteins attached to it. So RNA polymerase will get access to the promoter and it can initiate the process. When we learned about transcription, we learned that initiation process, how it is taking place. DNA dependent RNA polymerase will bind to the promoter in presence of an initiation factor and the process starts. Okay, Nandana? But here, when lactose molecule is not there, there is nobody to inactivate the repressor protein. The repressor protein will be produced and there is nobody to monitor it. This repressor protein will bind with the operator. And once repressor binds with operator, the RNA polymerase enzyme cannot attach to the promoter. If the transcription process should take place, it should bind with RNA polymerase, should bind with promoter. But the repressor protein will prevent RNA polymerase from binding to the promoter, and thereby the transcription of remaining genes Z, Y, A cannot take place. So in the absence of these enzymes, the metabolism of lactose also cannot be carried out. But when lactose is there, what difference it makes? The lactose, there also the repressor protein is formed. But after the formation of the repressor protein, our inducer or lactose will go and bind with the repressor and inactivates it. Once inactivated, they cannot bind with the operator. So if it cannot bind there, then who will get access? RNA polymerase, and the process can happen. Nandana, are you getting? Have you followed? You message me. If any more doubts are there, I will explain. No problem. Message me if any. Others also. If you are hesitant to say, please message me. Okay, clear no, Nandana? Yes. And you had doubts regarding elongation of transcription? Elongation stage of transcription? Correct? Yes. Just a minute. I was a bit fast, so it's not responding. Yes. Nandana, this stage, elongation. The first process, like initiation, you have understood, no, Nandana? Nandana, are you there? Elongation. Hope you have understood the first step. Initiation, elongation, termination. So I'll just show you the previous slide also. Three steps: initiation. In initiation, the RNA polymerase should bind with promoter. Only then the transcription process can start. And how they can recognize the promoter? With the help of an initiation factor or sigma factor. So RNA polymerase will associate with initiation factor. Together they will bind to the promoter and the process will begin. And that is how the process starts. Okay. Now next is elongation. Now once it is started, we need the synthesis. We need to synthesize that messenger RNA. And how that process is taking place? How it is taking place? RNA polymerase, where it has got, first it will attach to the promoter. You can see in the figure, it is getting attached to the promoter region with the help of initiation factor. Once it has started or it is bound to the promoter region, the next step is unzipping the DNA double helix. See, here you can see the unzipping has taken place. The two complementary strands which were lying close together, it unzipped, it got separated, and it will form an open loop. You can see in the figure that open loop over there. So this is the open loop. It has got unzipped and an open loop is formed. It uses ribonucleic because we have to synthesize RNA. While synthesizing DNA, we made use of deoxyribonucleic. Why, Nandana? Yes, not this one. Ah, she's here. Uh-huh. I was dying to explain. You missed the class? Shall I? You heard, no? From here, shall I repeat again? You message me if something is there. I didn't know. Oh, translation. Okay. I asked you whether it is transcription. Okay, no problem. This you had understood, Nandana? This is clear. Nandana, have you understood? Where are you? Turn your videos on. Okay, Nandana, no problem. I'll repeat it again tomorrow. Okay, we have a period tomorrow, and the translation process once again I will repeat. Maybe others are hesitant to say, they also might be having the same doubts. I'll repeat it again tomorrow, no problem. Okay, but somewhat 80% have you understood, Nandana? Nandana, tell me in percentage how much you understood. Okay, good. 90%. Okay, thank you, children. See you next class. Tomorrow we have a period I will arrange. Mostly it will be fourth period. I will let you know. Okay, all should join by tomorrow, and day after tomorrow we'll be winding up the portions. And very important topic tomorrow as well as day after tomorrow. Please don't miss the classes. So thank you.